Hello everyone and welcome back to yet another video of biology series. For the ones who new here, we are doing NCRT, every line of it, and this can be like a quick audiobook and also with the NCRT highlights. So you can do this at the last moment and this can be something you can do on the go. So without further ado, let's get started. In the previous video, we had actually done the first part of molecular basis of inheritance and we'll be taking that forward up. So yeah. A gene is defined as the functional unit of inheritance. Though there's no ambiguity that the genes are located on the DNA, it is difficult to literally define a gene in terms of DNA sequence. So we already know that genes are present on the DNA. We know this from ever since, but we actually have to define it in terms of DNA, like genes actually kahan pe present hai. The DNA sequence coding for tRNA and rRNA molecule also define a gene. However, by defining a cistron as a segment of DNA coding for a polypeptide, the structural gene in a transcription unit could be said as monocistronic, mostly in eukaryotes, or polycistronic, that is in bacteria or eukaryotes. So what happens basically is, in the DNA sequence, which is coding for a certain gene, alright, it is coding for a certain amino acid, for a certain polypeptide chain, you will have some units of the DNA that's going to transcribe up, fine. And cistron, the term cistron over here again very important, but this is basically a segment of DNA, fine, जो कि एक particular polypeptide के लिए code करेगा, fine. And this structural gene in a transcription unit, ये monocistronic भी हो सकता है, यानी एक ही cistron होगा, there'll only be one structural gene, or it can be polycistronic, fine. In eukaryotes, the monocistronic structural genes have interrupted coding sequences. The genes in eukaryotes are split. This is very important. The split gene system in case of eukaryotes. Eukaryotes में भी genes होते हैं, but they're not like a continuous line on the DNA, but they're split. The coding sequences or express sequences are defined as exons. जो express होंगे, जो code करेंगे, उनको exons बोलेंगे. Exons are said to be those sequences that appear in mature or processed RNA. The exons are interrupted by introns. Introns or intervening sequences do not appear in mature or processed RNA. ठीक है, mature RNA में, processed RNA में introns नहीं होते क्योंकि introns code नहीं करते, fine. So they like this split gene arrangement further complicates the definition of a gene in terms of a DNA segment. तो ऐसा नहीं है ना, एक gene है और उसके ऊपर हमने एक DNA है, उसके ऊपर एक gene का ऐसे line लगा दे. No, in case of eukaryotes, the cistrons even they will be although it's mostly monocistronic, but it will be split. Inheritance of a character is also affected by promoter and regulatory sequences of a structural gene. Hence, sometimes the regulatory sequence are loosely defined as regulatory genes, even though these sequences do not code for any RNA or protein. अब in this particular paragraph, they're trying to say कि जो हमारे पास एक transcription unit होता है, उसमें भी हमारे पास, for example, you have promoter, you have regulatory sequences, वो कोई भी ऐसे, they're not cistrons, they do not code for a particular RNA or protein, but yet we call them sometimes the regulatory genes, fine. Why? Because the necessity for sequencing, आगे जाके जो regulatory sequences से promoter इन्हें दिया हुआ है, they are also important for transcription. Types of RNA and the process of transcription. In bacteria, there are three major types of RNA. We have mRNA, we have tRNA, and we have rRNA. We know it, all three RNAs are needed to synthesize a protein in a cell. The mRNA provides the template, tRNA brings the amino acids and reads the genetic code, and rRNA plays structural and catalytic role during translation. So all will have their own function. जो हमारे पास messenger RNA है, that will provide the template, fine, and the transfer RNA will bring amino acids जिनको polypeptide बनाना है, and it will read the genetic code, what kinds of amino acids are to be brought. और जो हमारे पास ribosomal RNAs है, they also have some catalytic role. This single DNA dependent RNA polymerase that catalyzes transcription of all types of RNA in bacteria. RNA polymerase binds to promoter and initiates transcription. उसको हम initiation बोलते हैं. RNA polymerase हमारा enzyme है, वो promoter के साथ bind करके, we know promoter will be now in the transcription unit of the DNA. Promoter के साथ bind करके will initiate transcription. Fine. It uses nucleoside triphosphates as the substrate. So this is the promoter region, this is the RNA polymerase binding to it. And for the binding part, you need a sigma factor. Fine, this is the DNA helix. Elongation again, you have this, ये आगे आगे बढ़ रहा है, and then you'll have a terminator region जहाँ पे termination होगा, fine. On termination के लिए you need a rho factor. Let's see what they're trying to write. And polymerizes in a template dependent fashion following the rule of complementarity. Obviously हर चीज में complementarity आ गया. It somehow also facilitates opening of the helix and continuous elongation. Only a short stretch of RNA remains bound to the enzyme. Once the polymerase reaches the terminator region, the nascent RNA falls off. So also the RNA polymerase, this results in the termination of transcription. So what happens is basically हमारे पास एक stretch of RNA जो है सिर्फ वही एंजाइम के साथ जुड़ा हुआ होता है, ठीक है, और जो एक दफा पॉलीमरेज हमारा जो एंजाइम है, वंस इट रीचेस द टर्मिनेटर रीजन, आरएनए जो हमारा इट विल फॉल ऑफ, ठीक है, तो जो नेसेंट आरएनए है वो गिर जाएगा, फाइन, और आरएनए पॉलीमरेज भी. An intriguing question is that how is the RNA polymerase able to catalyze all the three steps, which are initiation, elongation, and termination, in a single enzyme? The RNA polymerase is only capable of catalyzing the process of elongation. You have to remember, जो हमारा RNA polymerase है वो सिर्फ elongation ही करवा सकता है. It associates transiently with initiation factor and termination factor to initiate and terminate the transcription respectively. तो ये जो हमारे पास initiation factors है, हमारे पास termination factors है, वो क्यों ही आए इधर? Why? Because the single RNA polymerase is not capable of catalyzing all the processes. It will only catalyze the process of elongation. Fine. Association with these factors alter the specificity of the RNA polymerase to either initiate or terminate. Fine. In bacteria, since the mRNA does not require any processing to become active, and also since transcription and translation take place in the same compartment, so there's no separation of cytosol and nucleus in the bacteria. Why? Because it's prokaryotic, right? तो हमारे पास कोई demarcation नहीं है कि भाई ये cell है, ये nucleus है. No, simply directly mRNA, उसको कोई processing की जरूरत नहीं. Directly it'll be active and it'll directly transcribe into proteins and translate into proteins. Fine. Many times the translation can begin much before the mRNA is fully transcribed. Consequently, the transcription and translation can be coupled in bacteria. Fine, ये दोनों simultaneously भी हो सकते हैं because ऐसा कोई खास demarcation ही नहीं है. In eukaryotes now there are two additional complexities. Eukaryotes में वैसा कुछ नहीं. There are at least 3 RNA polymerase in the nucleus in addition to the RNA polymerase found in the organelles. एक तो organelles में अलग है, उसके addition में nucleus में 3 RNA polymerase है. There's a clear cut division of labor. The RNA polymerase first transcribes ribosomal RNAs. अब हमारे पास एक clear cut demarcation होने वाला है कि certain enzymes are going to transcribe for certain ribosomes only. Over here we have, yes, RNA, अच्छा 28S, 18S, 5.8S, ये सारे हमारे ribosomal RNAs है. Whereas the RNA polymerase third is responsible for the transcription of tRNA, 5S RNA, and snRNA. This is basically small nuclear RNA. These are very important for numericals. You just have to remember. I cannot make you learn this. You have to learn it for yourself. The RNA polymerase second, then you have second है बीच वाला है, transcribes precursor of mRNA. What is the precursor of mRNA? It's the heterogeneous nuclear RNA, which is the hnRNA. Fine. The second complexity is that the primary transcripts contain both the exons and the introns and are non-functional. Hence, it is subjected to a process of splicing where the introns are removed and exons are joined in a defined order. Heterogeneous nuclear RNA undergoing additional processing called as capping and tailing. In capping, an unusual nucleotide, methyl guanosine triphosphate, is added to 5' of hnRNA. In tailing, adenylate residues 2 to 300 are added at 3' in a template independent manner. It is the fully processed hnRNA, now called mRNA, that is transported out of the nucleus for translation. So basically, in case of eukaryotes, complexity बढ़ गया है. अब directly एक तो पहले हमारे पास different RNA polymerase होंगे, different जो हमारे प्रोसेसेस है उनको कैटलाइज करने के लिए. अब हमारे पास जो हमें ये भी पता है कि वहाँ पे you have split gene system in case of eukaryotes, so it's not just all the coding part of the genes, so you'll have introns and you'll have exons. So for that again you have to do splicing. The introns will be removed and the exons will be joined. Then for heterogeneous nuclear RNA, जिससे बाद में mRNA बनेगा, उसकी कैपिंग करनी है, टेलिंग करनी है, ठीक है. कोई unusual nucleotide capping is added to the 5' end. Fine. Tailing में adenylate residues are added at the 3' end. Moving on. The significance of such complexities is now beginning to be understood. The split gene arrangements represent probably an ancient feature of the genome. ये आया ही क्यों? The presence of introns is reminiscent of antiquity, and the process of splicing represents the dominance of RNA world. In recent times, the understanding of RNA and RNA dependent processes in the living system have assumed more importance. Right? तो क्यों हमारे पास usually we need RNA basically for splicing purposes, ठीक है? And why is it so थे ही क्यों split genes in the first place, right? It's a question. So we can say exons and splicing will make it possible to have a greater number of proteins made than there are genes, ठीक है, क्योंकि हमारे पास बहुत सारे splitting हो रही है, और जो introns बीच में आ रहे हैं, they'll enable alternative splicing which will enable a single gene to encode multiple proteins, fine, जिनके different functions होंगे different conditions में, fine. Moving on to the genetic code now. During replication and transcription, a nucleic acid was copied to form another nucleic acid. Hence, these processes are easy to conceptualize on the basis of complementarity. ये तो आप समझते हैं, complementary basis बनाएंगे, ठीक? इससे transcription भी होगा और इसी से replication भी होगा. The process of translation requires transfer of genetic information from a polymer of nucleotides to synthesize a polymer of amino acids. अब हमें क्या बनाना है? Nucleic acid से protein बनाना है. समझ आ रही है? Neither does any complementarity exist between nucleotides and amino acids, nor could any be drawn theoretically. They're two different entities. There existed ample evidences, though, to support the notion that change in nucleic acids or genetic material were responsible for change in amino acid and proteins. This led to the proposition of a genetic code that could direct the sequence of amino acids during synthesis of proteins. अब, however, क्या हुआ? There was this thought कि usually जब कोई change nucleic acid में आएगा, that will be reflected in the proteins. So they were trying to draw, the scientists were trying to draw a relationship between the amino acid sequence and the nucleotide sequence. So there's a certain code that is going to express itself as a certain amino acid. वही हम यहाँ आगे देखने वाले हैं. If determining the biochemical nature of genetic material and the structure of DNA was very exciting, the proposition and deciphering of genetic code was most challenging. In a very true sense, it required involvement of scientists from several disciplines: physicists, organic chemists, biochemists, geneticists. It was George Gamow, a physicist, who argued that since there are only 4 bases and if they have to code for 20 amino acids, 4 bases है और हमारे पास 20 amino acid है. bases यानी 4 हमारे पास nitrogen bases होते हैं ना, that's what differs in a nucleotide, right? बाकी तो सारा phosphate और sugar is same, fine. So 4 bases है, purines 2 है, pyrimidines है. इनमें क्या हो रहा है? They have to code for 20 amino acids, so the code should constitute a common. So he used basically permutation combination. He suggested that in order to code for all the 20 amino acids, the code should be made up of 3 nucleotides. This was a very bold proposition. So it was Gamow who gave the genetic code first. He said that 20 amino acid है और उनको code करने के लिए हमारे पास 4 bases है. हम उनको किस तरीके से 4 का cube, you will get 64 codons, you will get 64 triplets because 3 nucleotides एक amino acid के लिए code करेंगे. Fine, generating many more codons than required. Providing proof that the codon was a triplet was a more daunting task. The chemical method developed by Hargobind Khurana, now experimentally providing a proof कि जो codon है हमारे पास वो एक triplet ही था. Even this has been asked. It was Hargobind Khurana. ठीक है? उसने क्या किया? A chemical method developed by him was instrumental in synthesizing RNA molecules with defining combinations of bases, homopolymers and copolymers. Marshall Nirenberg's cell-free system for protein synthesis finally helped the code to be deciphered. So finally it was Nirenberg जिसके protein synthesis ने हमें बताया कि code हम कैसे लाएंगे. Severo-Ochoa enzyme polynucleotide phosphorylase जो होता है was also helpful in polymerizing RNA with defined sequences in a template independent manner. कोई template नहीं था, you could simply produce RNA. Enzymatic synthesis of RNA उसको बोलते हैं. So this is the enzyme. ठीक है, Severo-Ochoa enzyme जिससे हम RNA synthesize कर रहे थे. Finally, a checkerboard for genetic code was prepared, which is given over here. Now you can remember as many as possible. It can come in the exam, it cannot. ठीक है, you can just try and just remember. ये हमारे पास triplet codons दिए हुए हैं, ठीक है, bases का 3-3 के arrangement में, और हमारे पास बन रहे हैं almost 20 amino acids जो हमें ज़रूरत है. Fine. अब इसके क्या है, जेनेटिक कोड के क्या ही हमारे पास यहाँ पे फीचर्स है. लेट्स गो थ्रू देम. द कोडॉन इज अ ट्रिपलेट, ये तो हमें समझ आ गया. 61 कोडॉन्स कोड फॉर अमीनो एसिड्स एंड 3 कोडॉन्स डू नॉट कोड फॉर एनी अमीनो एसिड, हेंस दे फंक्शन एज स्टॉप कोडॉन्स. हमारे पास 64 है टोटल में, 4 into 4 into 4. फाइन. 64 में से 61 जो है दे विल कोड फॉर अमीनो एसिड, 3 विल नॉट कोड. ठीक. सम अमीनो एसिड्स आर कोडेड बाय मोर देन 1 कोडॉन, हेंस द कोड इज डीजेनरेट. कुछ अमीनो एसिड्स, क्योंकि हमारे पास बहुत सारे कोडॉन्स है, ठीक है, और अमीनो एसिड सिर्फ 20 ही है, तो कुछ अमीनो एसिड्स जो है बहुत सारे कोडॉन से वो कोड होते हैं. तो कोडॉन इज रेड इन mRNA in a continuous fashion. There are no punctuations. ऊपर भी देखा होगा आपने, the code is nearly universal. From bacterium to human, you would code for phenylalanine. ये तो बहुत easy है, ये तो याद रख ही सकते हैं. Some exceptions to this rule have been found in mitochondrial codons and in some proteins. AUG has dual functions, very important. AUG mostly, it codes for methionine, and if it will also act as the initiator codon. अगर ये initiation place पर है तो initiator codon है, but it will also code for methionine. UAA, UAG and UGA are stop terminator codon, so they do not code for any amino acid. If following is the sequence of nucleotides in mRNA, predict the sequence of amino acid and coded by it, and you can take the help of. अच्छा, इसके देखने हैं आपको, this is methionine, ऊपर ही हमने देखा this is phenylalanine वगैरह, ठीक है. So you can just do it from the code given above. You can try and find out कि हमारे पास कौन सा polymer chain बन के आएगा. Now try the opposite. अब यहाँ पे सीक्वेंस दिया हुआ है अमीनो एसिड्स का, इससे आप डिराइव करेंगे हमारा दिस विल स्टार्ट विद AUG एंड स्टफ, ठीक. डू यू फेस एनी डिफिकल्टी इन प्रिडिक्टिंग द ऑपोजिट? नाउ कमिंग टू म्यूटेशंस एंड जेनेटिक कोड. The relationship between gene and DNA are best understood by mutation studies. अभी भी हम रिलेशन ही पढ़ रहे हैं DNA और जीन के बीच में. You've studied about mutation. Effects of large deletions and rearrangements in a segment of DNA are easy to comprehend. It may result in loss or gain of a gene and so a function. The effect of point mutations will be explained here. A classical example of point mutation is a change of single base pair in the gene for beta globin chain that results in the chain of amino acid residue glutamate to valine. It results into a diseased condition called as sickle cell anemia. Effect of point mutations that inserts or deletes a base in structural gene can be better understood by following simple example. अब यहाँ पे क्या दिया हुआ है, इन्हें बोला हमने mutations already पढ़े हैं. अगर हमारे पास कहीं DNA में कहीं genetic material में rearrangement होता है, it can be lethal because mutations usually harmful होते हैं. Fine. अब जो है हमारे पास ये point mutation का एक example हमने देखा था कि अगर हमारे पास ग्लूटामेट जो है उसकी जगह पे वैलीन आ जाता है तो हमारे पास सिकल सेल एनीमिया होता है. फाइन. इन द बीटा ग्लोबिन चेन इनसाइड अ ह्यूमन बीइंग. फाइन. तो दिस वाज एन एग्जांपल ऑफ अ पॉइंट म्यूटेशन. कंसीडर अ स्टेटमेंट दैट इज मेड अप ऑफ द फॉलोइंग वर्ड्स ईच हैविंग 3 लेटर्स लाइक अ जेनेटिक कोड. ये कुछ भी है. रैम हैज रेड कैप. इफ वी इंसर्ट अ लेटर बी इन बिटवीन हैज एंड रेड एंड रीअरेंज द स्टेटमेंट, इट वुड बी RAM has pre-DCAP. Now can you see you cannot read it properly? Why? क्योंकि हम लोग क्या करें basically, we are inserting letter B in between has and read. फाइन. अब ये रीअरेंज हो जाएगा बिकॉज इसको आगे आगे चलना है. कोड ऑन तो 3 का ही होगा. सिमिलरली वी नाउ इंसर्ट द लेटर्स 2 लेटर्स एट द सेम प्लेस. से BI. नाउ इट वुड रीड लाइक दिस. अगर हम 3 लेटर्स बिग ही करें देन द स्टेटमेंट वुड रीड लाइक दिस. ठीक. तो आगे का तो हमारा सारा सेम रहा. इन केस ऑफ दिस एंटायर, इफ यू पुट एन एंटायर कोड ऑन, द सेम एक्सरसाइज कैन बी रिपीटेड बाय डिलीटिंग द लेटर्स. ठीक. अब आप डिलीट कर सकते हैं. 1 डिलीट किया, 2 डिलीट किए, फिर तीनों डिलीट किए. फाइन. This is what they're trying to do. Mutate कर रहे हैं. ठीक. The conclusion from the above exercise is very obvious. Insertion or deletion of 1 or 2 bases changes the reading frame from the point of insertion or deletion. However, such mutations are referred to as frame shift or deletion mutations. यानी अगर आपने 1 या 2 जो है हमारे points उधर पे हमने change लाया. Fine. Insertion or deletion of 1 or 2 bases. Only 1 or 2, not 3. तो फिर पूरा codon change हो जाएगा. अगर हमने 1 या 2 base change किया उसकी वजह से क्या हो जा रहा है, हमारा frame shift, insertion और deletion mutation. What is that basically? That is कि हम जो उसके आगे से if we try to read the sequence, it will not read the same as previously we have read it. Insertion or deletion of 3 or its multiple bases, insert or delete in 1 or multiple codon, hence 1 or multiple amino acids, and reading frame remains unaltered from that point onwards. अगर हमने multiple bases ही add किए, मतलब हमने पूरा का पूरा triplet codon ही add कर लिया, तो उसके आगे क्या होगा, reading will remain same, it will remain unaltered. वो आपको ऊपर भी समझ आया होगा. Let's come to tRNA, the adapter molecule. From the very beginning of the proposition of code, it was clear to Francis Crick that there has to be a mechanism to read the code and also to link to the amino acids, because amino acids have no structural specialities to read the code uniquely. Amino acids के पास ऐसा कुछ भी नहीं है कि वो read कर सकते हैं code और वो खुद ही खुद को replicate करेंगे या वो खुद ही polymer बनाएंगे. ऐसा कुछ नहीं है. He postulated the presence of an adapter molecule that would on one hand read the code and on other hand would bind to specific amino acid. अब उसने बोला कि हमारे पास एक मॉलिक्यूल ऐसा आएगा, एक अडेप्टर मॉलिक्यूल होगा जो एक हाथ से तो कोड रीड करेगा और उसको समझ आएगा कौन सा अमीनो एसिड लाना है, और दूसरी तरफ से इट इज गोइंग टू बाइंड टू द अमीनो एसिड एंड इवेंचुअली फॉर्मुलेट अ चेन. द tRNA, then called sRNA, soluble RNA, was known before the genetic code was postulated. However, its role as an adapter molecule was assigned much later. पता हमें पहले था बट फंक्शन नहीं पता. tRNA has an anticodon loop. That is basis complementary to the code. तो ये हमारे पास एंटीकोडॉन लूप हो गया और ये किसके साथ कॉम्प्लीमेंटैरिटी दिखाएगा, विद द कोडॉन जो इसको कोड करना है. फाइन. ये हमारा mRNA, इस पे से इसने देखा इस कोडॉन को और इसके पास इसी कोडॉन के एंटीकोडॉन्स होंगे. and it also has an amino acid acceptor end to which it binds to amino acids. यहाँ पे हमारे पास होगा amino acid acceptor end. tRNAs are specific for each amino acid. For initiation there's another specific tRNA that is referred to as initiator tRNA. ठीक है, हमारे पास initiator tRNA भी होगा जो initiate करेगा polymerization of the amino acids into a polypeptide chain. There are no tRNAs for stop codons. Very important. Stop codons के लिए कोई भी tRNA नहीं है क्योंकि वो they do not code for any amino acid. The secondary structure of tRNA has been depicted that looks like a clover leaf. ठीक है, in actual structure the tRNA is a compact molecule which looks like inverted L. तो अगर जब आप इसको 3D में देखते हैं ना, it's like an inverted L. But you also have the clover leaf model which was given by Hawley. Fine. Coming to translation. Translation refers to the process of polymerization of amino acids to form a polypeptide. The order and sequence of amino acids are defined by the sequence of bases in the mRNA. mRNA के पे जो bases होंगे उन्हीं की sequence that is going to code for a certain polypeptide. The amino acid are joined by a bond which is known as the peptide bond. ये तो आपने chemistry में भी किया होगा. Formation of a peptide bond requires energy. Of course bond बनना है तो energy चाहिए. Therefore in the first phase itself amino acids are activated in the presence of ATP and linked to their cognate tRNA, a process commonly called as charging of tRNA or amino acylation of tRNA to be more specific. तो सबसे पहले क्या होता है, हमारे पास in the presence of ATP जो हमारा amino acid है that will get linked to its specific tRNA. इसको हम charging of tRNA बोलते हैं या amino acylation of tRNA बोलते हैं. If two such charged tRNAs are brought close enough, the formation of peptide bond between them would be favored energetically. The presence of a catalyst would enhance the rate of peptide bond formation. समझ आई? दो tRNAs को अगर आप साथ में लाते हैं, so they have a tendency to get combined and they'll form the polypeptide chain. Fine? ये हमारा एक tRNA है. जब ये बाद में चला जाएगा, यहाँ पे दूसरा tRNA आ जाएगा, and what is it trying to do? ये इस पे attach हो जाएगा. Fine? This entire thing will get attached to this. The cellular factory responsible for synthesizing proteins is the ribosome. हमें पहले भी पता ribosome है ही क्यों, there for protein synthesis. The ribosome consists of structural RNAs and about 80 different proteins. In its inactive state, it exists as two subunits, a large subunit and a small subunit. बनते का eventually हमारे ribosomes. हाँ, हमारे पास एक large subunit होता है और एक small subunit होता है ribosome का, और वहाँ पे हमारे proteins बनते हैं. Alright, when the small, अब inactivity में दो subunits होंगे. अब क्या होगा, when the small subunit encounters an mRNA, the process of translation of the mRNA to protein begins. Small subunit comes in contact with the mRNA and the process of translation will begin. There are two sites in the large subunit. Large subunit में दो sites है for subsequent amino acids to bind and thus be close enough to each other for the formation of a peptide bond. ठीक है, large subunit में दो sites है जो बहुत नजदीक होती हैं, which are going to facilitate the binding of the two amino acids on two different tRNAs. Fine. The ribosome also acts as a catalyst. Ribosome पे जो 23S rRNA in bacteria is the enzyme ribozyme. ऐसे ribosomes जो enzymes की तरह काम करें उनको हम ribozyme बोलते हैं. Fine. For the formation of peptide bond. Fine. So that is how 23S rRNA जो है वो bacteria में it is the ribozyme and it will help for the formation of the peptide bond. A translational unit in mRNA is the sequence of RNA that is flanked by the start codon AUG and the stop codon and codes for a polypeptide. Fine, तो एक translational unit होगा. Transcriptional unit हमारे DNA पे होता था, translational unit mRNA पे होगा. Fine. An mRNA also has some additional sequences that are not translated and are referred to as untranslated regions. UTRs are untranslated regions जो mRNA पे ही होते हैं. They're present at both 5' before the start codon and 3' after the stop codon. Fine, they're required for efficient translation processes. So there are certain breaks जिनको untranslated regions बोलते हैं. 5' के पहले होगा और क्या होगा 3' के बाद होगा. Fine. Moving on, we have for initiation, the ribosome binds to the mRNA at the start codon. Start codon हमें पता है AUG coding for methionine as well. That is recognized only by the initiator tRNA. Fine. Initiate कहाँ से होता है? Ribosome mRNA को bind हुआ, ठीक है, कहाँ पे bind हुआ? Start codon पे bind हुआ, और ये किसने recognize किया? Initiator tRNA ने. The ribosome proceeds to the elongation phase of protein synthesis. During this stage, complex is composed of an amino acid linked to tRNA. Sequentially bind to the appropriate codon in mRNA by forming complementary base pairs with the tRNA anticodon. तो अब क्या होगा? Consequently आगे आगे the chain will forward. Fine, move forward. और tRNA जो है it will bind to the appropriate codon in mRNA कॉम्प्लीमेंट्री बेसिस बनाएगा बिकॉज टीआरएनए के पास एंटी कोडॉन्स है. द राइबोसोम मूव्स फ्रॉम कोडॉन टू कोडॉन अलोंग द एमआरएनए. अमीनो एसिड्स आर एडेड वन बाय वन, ट्रांसलेटेड इनटू पॉलीपेप्टाइड sequences dictated by DNA and represented by mRNA. It's a very important line. Start कहाँ से हुआ? Dictation DNA का ही था, लेकिन represent किसने किया? Express बाद में जाके किसकी वजह से हुआ? mRNA की वजह से. At the end, a release factor. There's a release factor which binds to the stop codon, terminating translation and releasing the complete polypeptide from the ribosome. तो release factor है because हमारे पास कोई stop anticodons वाला कोई tRNA नहीं होता, so you have the release factor binding to the stop codon which will terminate the translation. Next up, we have regulation of gene expression. Regulation of gene expression refers to a very broad term that may occur at various levels. Considering the gene expression results in the formation of a polypeptide, it can be regulated at several levels. In eukaryotes, the regulation could be exerted at the transcription level, processing level, transport of mRNA from nucleus to the cytoplasm, and translational level. So basically, regulation क्या है कि हमें अगर we need a certain result, you need a certain polypeptide, you can regulate it. You can choose what polypeptide you can form, and this can happen at different levels. ठीक? The genes in a cell are expressed to perform a particular function or a set of functions. For example, if an enzyme called beta-galactosidase is synthesized by E. coli, it is used to catalyze the hydrolysis of a disaccharide lactose into galactose and glucose. We know this. अगर हमारे पास एक enzyme है beta-galactosidase, this enzyme ये किस लिए होता है वहाँ? It's for the hydrolysis of lactose, जिसको बाद में galactose और glucose में बनाता है. The bacteria use them as a source of energy. Hence, if the bacteria do not have lactose around them to be utilized for energy source, they would no longer require the synthesis of the enzyme beta-galactosidase. Just straight up facts, we know that अगर lactose ही नहीं है, उसको hydrolyze ही नहीं करना है, तो कोई enzyme बनाने की ज़रूरत नहीं है. Therefore, in simple terms, it is the metabolic, physiological, environmental conditions that regulate the expression of genes. Gene बनना है, express होना है या नहीं होना है. Basically, it depends upon various factors. The development and differentiation of embryo into adult organisms are also a result of the coordinated regulation of expression of several sets of genes. Fine. इसी तरह जब हमारा embryo जो है differentiate होके fetal body में convert हो जाता है, उसमें भी you need to have a regulation of several set of gene. मतलब वो कैसे खुद को express करेंगे. In prokaryotes, control of the rate of transcriptional initiation is the predominant site for control of gene expression. अब eukaryotes में तो बहुत सारे sites होते हैं. Prokaryotes में usually जहाँ पे transcription initiate होता है वहाँ पे आप gene expression को control कर सकते हैं. In a transcription unit, the activity of RNA polymerase at a given promoter is in turn regulated by interaction with accessory proteins, which affect its ability to recognize start sites. ठीक है. RNA polymerase जो होता है वो एक promoter region में क्या करता है, वो regulate करता है interaction with accessory proteins जो बाद में to recognize the start site. Fine. If you need to have an RNA polymerase which is bound to the promoter region. The accessibility alright, these regulatory proteins can act both positively as activators and negatively as repressors. ठीक है, तो हमारे पास activation भी हो सकता है, repression भी हो सकता है based upon हमें क्या चाहिए, what is the requirement. The accessibility of promoter regions of prokaryotic DNA, as in many cases, regulated by the interaction of proteins with sequences termed as operators. ठीक है. तो हम देखते हैं यूजुअली प्रोकैरियोट्स में ऐसी कौन सी चीज़ है जो ऑपरेट करती है, जो बेसिकली हमें हेल्प करती है रेगुलेशन में. वी हैव ऑपरेटर्स. ऑपरेटर्स क्या होते हैं? Proteins will bind to sequences in a certain way that they're going to regulate whether gene is going to be expressed or not. The operator region is adjacent to the promoter elements. Operator भी promoter के साथ में ही होता है. In most operons and in most cases, the sequences of the operator bind a repressor protein. Fine. Usually repression के लिए. Each operon has its specific operator and specific repressor. Fine. So specificity आ रही है. For example, lac operator is present only in the lac operon and it interacts specifically with lac repressor. Only we're going to actually do the lac operon concept right now. This is again an example of gene expression, how you're controlling it, right? The elucidation of the lac operon was also a result of a close association between a geneticist, Francis Jacob, and biochemist Jake Monod. So I think this also you have to remember, Jacob and Monod were the ones जिन्होंने lac operon का concept दिया. They were the first to elucidate a transcriptionally regulated system. Regulation कर रहे हैं transcription में. You're regulating what genes are to be expressed and what are not to be expressed, fine? In lac operon, here lac will refer to lactose, a polycistronic structural gene जिसमें बहुत सारे cistrons है, is regulated by a common promoter and regulatory genes. Such arrangement is very common in bacteria and is referred to as an operon. ठीक है, एक हमारे पास polycistronic structural gene है, एक promoter region है और regulatory genes है, और ऐसे arrangement को हम operon बोलते हैं. To name few examples, lac operon, trip operon, ara, his, val, etc. Fine, which are there for different substances. The lac operon consists of one regulatory gene, the I gene. Here the term I does not refer to inducer, rather it is derived from the word inhibitor. So, and you have three structural genes, Z, Y and A. हमारे तीन structural genes है, और एक regulatory gene है जिसको हम I gene बोलते हैं. I का मतलब यहाँ पे inhibitor है. The I gene, ये क्या है basically? This is a regulatory gene. It will code for the repressor of the lac operon. ठीक है, क्योंकि इनहिबिट कर रहा है. द Z gene codes for beta galactosidase, which is primarily responsible for the hydrolysis of the disaccharide lactose into its monomeric units. ये हमें पता है, बीटा गैलेक्टोसिडेज क्या करेगा, गैलेक्टोज और ग्लूकोज में तोड़ेगा लैक्टोज को. द Y gene codes for permease. Y gene is coding for permease, which increases the permeability of the cell to beta galactosidases. The A genes, अब A gene क्या है, they encode a trans acetylase. Hence all the three gene products in lac operon are required for the metabolism of lactose. ये हम एक certain transcriptional unit ले रहे हैं यहाँ पे, जिसमें हम lactose metabolism देख रहे हैं. In most other operons as well, the genes present in the operon are needed together to function in the same or related metabolic pathway. So they have to work in a coordinated way in order to metabolize the lactose that is given. अब यहाँ पे आप will actually first read it out first. Lactose is the substrate for the enzyme beta galactosidase and it regulates switching on and off of the operon. Hence it is named as inducer. तो inducer हमारा खुद से lactose ही होगा, वो जब present होगा तो enzyme बनेगा. In the absence of a preferred carbon source such as glucose, if lactose is provided in the growth medium of the bacteria, the lactose is transported into the cells through the action of permease. अगर glucose नहीं है तो उसकी beta galactosidase अंदर लेने की जो permeability है वो बढ़ जाएगी, और वो permease की वजह से होती है, lactose अंदर लेने की. Because अगर ग्लूकोज नहीं है तो हम लैक्टोज से ही अपना काम चलाएंगे. Remember, a very low level of expression of lac operon has to be present in the cell all the time, otherwise lactose cannot enter the cells. ठीक है, the lactose then induces the operon in the following. अब induction कैसे होगा, हमारे पास अब ये समझ आ गया कि lactose is present और it is going to switch on or off all of the regulation of the gene. Fine. The repressor of the operon is synthesized all the time constitutively from the I gene. Repressor कहाँ से आ रहा है, I gene से, inhibitor gene. The repressor protein binds to the operator region of the operon and prevents RNA polymerase from transcribing the operon. क्या हो रहा है basically, repressor जो है हर time present होता है वहाँ पे, आई जीन से निकलता है और ये बाइंड करेगा ऑपरेटर रीजन पे. ठीक, अब जब ऑपरेटर ही बंद हो गया, बाइंड हो गया आई जीन की वजह से, इनहिबिटर जीन की वजह से, तो आरएनए पॉलीमरेज विल नॉट एक्ट एंड देयर बी नो ट्रांसक्रिप्शन इन द प्रेजेंस ऑफ एन इंड्यूसर. अब अगर हमारे पास इंड्यूसर है, लैक्टोज ले लिया, एलोलैक्टोज ले लिया, द रिप्रेसर इज इनएक्टिवेटेड बाय इंटरेक्शन विद द इंड्यूसर. अब जब लैक्टोज है वो रिप्रेसर की वजह से क्या हो जाएगा, they are going to bind or eventually repressor will be inactivated. This allows RNA polymerase access to the promoter and transcription proceeds. Essentially, a regulation of lac operon can also be visualized as regulation of enzyme synthesis by its substrate. Remember, glucose or galactose cannot act as inducers for lac operon. क्यों? क्योंकि वो already broken products है, हमें उन्हीं को metabolize नहीं करना, हमें lactose को metabolize करना है. Fine. Regulation of lac operon by repressor is referred to as negative regulation. ठीक है, अब यहाँ पे क्या हो रहा है, lac operon repressor से क्या हो रहा है, it's regulating but in a negative sense, right? Lac operon is under control of positive regulation as well, but it is beyond the scope of the discussion at this level. Next we have the Human Genome Project. In the preceding sections, you've learned that it's the sequence of bases in DNA that determines the genetic information of a given organism. In other words, genetic makeup of an organism or an individual lies in the DNA sequences. If two individuals differ, then their DNA sequences should also be different, at least at some places. These assumptions led to the quest of finding out the complete DNA sequence of human genome. With the establishment of genetic engineering techniques where it was possible to isolate and clone any piece of DNA, and availability of simple and fast techniques for determining DNA sequences, a very ambitious project of sequencing human genome was launched in the year 1990. अब ये project आया ही क्यों? We thought that let's try out and assemble all of the genetic material of humans. हाँ, जब हमें पता है कि individual का जो भी properties है वो genetic material ही regulate करेगा, let's actually find out each and every base pair. Let's find out the sequences of the DNA inside the human genome. Fine. Human Genome Project was called a mega project अब आपको समझ आ रहा है, बिकॉज ऑब्वियसली हमारे अंदर बहुत सारा डीएनए, बहुत सारा जेनेटिक मटेरियल, उस सारे को अगर आपको सीक्वेंस करना है, इट इज अ ह्यूज प्रोजेक्ट. ह्यूमन जीनोम इज सेड टू अप्रॉक्सिमेटली 3 to 10 raised power 9 बेस पेयर, 3 बिलियन बेस पेयर्स का है. एंड इफ द कॉस्ट ऑफ सीक्वेंसेज इज रिक्वायर्ड इज $3 per base pair, the estimated cost in the beginning, the total estimated cost of the project would be approximately $9 billion US, right? Further, if the obtained sequences were to be stored in type form in books, and if each page of the book contained 1,000 letters and each book contained 1,000 pages, then 3,300 such books would be required to store the information of DNA sequence from a single human cell. The enormous amount of data expected to be generated also necessitated the use of high-speed computational devices for data storage and retrieval and analysis. HEPU is closely associated with the rapid development of a new area in biology called bioinformatics. अब यहाँ पे यही आपको अंदाजा दे रहा है, it's unimaginable to actually sequence all of the DNA. It requires a lot of expenses. It requires lots of space, lots of storage devices. तो बायोइन्फॉर्मेटिक्स भी इसने रिलेट किया. वाई? बिकॉज इट वॉज ट्राइंग टू स्टोर डेटा इन अ बेटर सेंस. कंप्यूटेशनल डिवाइसेस लिए बहुत सारे, फॉर ऑब्वियसली फॉर स्टोरेज पर्पसेस एंड stuff, because it's really difficult to assemble all of the DNA and all of the genome of the humans. Fine, that's why it was called a mega project. It was started in 1990. What are the goals now? Identify all the approximately 20 to 25,000 genes in human DNA. कितने genes हैं, उन सबको identify करेंगे. Determine the sequence of the 3 billion chemical base pairs that make up human DNA. Sequencing करेंगे 3 billion base pairs की. You'll store this information in data. Storage is also very important. पता करके फिर उसको कहीं store भी करना है. और store करना क्यों मुश्किल है? Because it's a huge amount of data. Improve tools for data analysis. Just I told you, we started to use computational devices and stuff. Transfer related technologies to other sectors such as industries. Address the ethical, legal and social issues, LC, ethical, legal and social issues that may arise from the project. The Human Genome Project was a 13-year project coordinated by the US Department of Energy and the National Institute of Health. Fine. During the early years of the HGP, the Wellcome Trust UK became a major partner. Additional contributions came from Japan, France, Germany, China and others. The project was completed in 2003. 13 साल लगे. 1990 में start हुआ, 2003 में खत्म हुआ. The knowledge about the effects of DNA variations among individuals can lead to revolutionary new ways to diagnose, treat and someday prevent the thousands of disorders that affect human beings. So ये भी इसका एक माने आप एक reason था that they wanted to sequence human genome ताकि वो पता कर सके जो genetic disorders होते हैं हम उनको कभी ट्रीट कर सकते हैं या नहीं. Besides providing clues to understanding human biology, learning about non-human organisms, DNA sequences can lead to an understanding of their natural capabilities that can be applied towards solving challenges in healthcare, agriculture, energy production, environmental remediation. ठीक है, ये सारे इसके ये थे consequences जो हम आगे जाके इससे मदद हासिल कर सकते हैं. Many non-human model organisms such as bacteria, yeast, Caenorhabditis elegans, a free-living non-pathogenic nematode, Drosophila, the fruit fly, plants, ricin, Arabidopsis, etc. have also been sequenced. इनका not just humans, we have sequenced DNA of certain other organisms. You have to remember these. Fine, bacteria, yeast, ये सब हो गए. And ricin and Arabidopsis is asked also, I guess, in AIPMT somewhere. Methodologies. कैसे किया? The methods involved two major approaches. One approach focused on identifying all the genes that are expressed as RNA, referred to as express sequence tags. अब ESTs क्या है? Basically, वो सारे genes जो RNA में express हो गए हैं, हम उनको focus करेंगे. That is express sequence tag. The other took the blind approach of simply sequencing the whole set of genome that contained all the coding and non-coding sequence and later assigning different regions in the sequence with functions. A term referred to as सीक्वेंस एनोटेशन. अब यहाँ पे क्या, या तो आपने सीधा डीएनए लिया, उसमें आपको एक्सॉन्स भी मिलेंगे, इंट्रॉन्स भी मिलेंगे, सही, उसको आपने फिर कोडिंग नॉन कोडिंग में डिफ्रेंशिएट करना है, उसको सीक्वेंस एनोटेशन बोलते हैं. या फिर आपने सीधा एमआरएनए देख लिया, एमआरएनए में भी वही जीन्स एक्सप्रेस हुए हैं, ठीक है, दैट इज एक्सप्रेस सीक्वेंस टैग्स. For sequencing, the total DNA from a cell is isolated and converted into random fragments of relatively smaller sizes. Recall, DNA is a very long polymer and there are technical limitations in sequencing very long pieces of DNA and cloned in suitable hosts using specialized vectors. तो हम क्या करें basically? DNA को isolate किया, उसको छोटे छोटे fragments में तोड़ा, and since it's a long polymer, so there are technical limitations in sequencing it. Fine. Then we cloned it. The cloning resulted into amplification of each piece of DNA fragment so that it subsequently could be sequenced with अगर एक ही फ्रैगमेंट के बहुत सारे कॉपीज हमारे पास होंगे, we can study it in a very better manner. The commonly used hosts were bacteria and yeast. तो कौन से host लिए? बैक्टीरिया और yeast. And the vectors were called as bacterial artificial chromosome and yeast artificial chromosome. The fragments were sequenced using automated DNA sequencers. ठीक है, हमारे पास कुछ automatic sequencers थे जो DNA को खुद ही sequence कर रहे थे. That worked on the principle of a method developed by Frederick Sanger. Sanger के method पे खुद ही sequence हो रहा था. Remember, Sanger is also credited for developing method for determination of amino acid sequences in proteins. So you have to remember Sanger, उसने क्या किया? DNA sequencing और amino acid sequencing में भी इसने help किया. Sequencing, S for sequencing, S for Sanger. You'll remember this forever. These sequences were then arranged based on some overlapping regions present in them. This required generation of overlapping fragments for sequencing. Alignment of these sequences was humanly not possible. हम align नहीं कर सकते हैं, ठीक है, but हम इनको देख रहे थे कि overlap कर रहे हैं over the generations. Therefore, specialized computer-based programs were developed. These sequences were subsequently annotated and were assigned to each chromosome. The sequence of chromosome 1 was completed only in May 2006. The last of the 24 human chromosomes was the first chromosome जो हमने recently sequence किया, and that was in 2006. Fine. This was all right. 22 autosomes and X and Y to be sequenced. Another challenging task was assigning the genetic and physical maps on the genome. This was generated using information on polymorphism of restriction endonuclease recognition sites and some repetitive DNA sequences known as microsatellites. कुछ repetitive DNA sequences थे, microsatellites. One of the applications of polymorphism in repetitive DNA sequences shall be explained in next section of DNA fingerprinting, जो हम आगे देखने वाले हैं. Fine. Salient features of human genome. The human genome contains 3164.7 million base pairs. वही 3 billion होता है around. The average gene consists of 3000 bases, but sizes vary greatly, with the largest known human gene being dystrophine at 2.4 million bases. The largest known human gene is dystrophine और उसके 2.4 million bases है. The total number of genes is estimated to be 30,000, much lower than previous estimates. ठीक. The functions are known for over 50% of the discovered genes. हमें पता है almost. Less than 2% of the genome codes for proteins. Less than 2%. Repeated sequences make a very large portion of the human genome. Repetitive sequences are stretches of DNA sequences that are repeated many times. Repetitive DNA sequences है, बार-बार repeat हो रहा है. Sometimes 100 to 1000 times. They're thought to have no direct coding functions, but they shed light on chromosome structure, dynamics, and evolution. Chromosome 1 has the most genes, 2968, and Y has the fewest. Very important. First chromosome के सबसे ज़्यादा genes है 2968. Y chromosome के सबसे कम है 231. Scientists have identified about 1.4 million locations where single base DNA differences, SNP, single nucleotide polymorphism, ठीक है, ऐसे 1.4 million locations है जहाँ पे humans में, you also pronounce it as SNPs, single nucleotide polymorphism. This information promises to revolutionize the process of finding chromosomal locations for disease-associated sequences and tracing human history. So basically, each single nucleotide polymorphism, it is going to represent a difference in a single DNA building block called a nucleotide. We know that. तो क्या होता है basically DNA में एक single point पे difference होना, एक single point पे variation होना, उसी को हम single nucleotide polymorphism बोलते हैं, एक base में difference होना. Fine. Applications and future challenges. Deriving meaningful knowledge from the DNA sequences will define research through the coming decades, leading to our understanding of biological systems. Obviously, हमारा advancement होगा, developing, and then, biological systems we can understand in a better sense. This enormous task will require the expertise and creativity of tens of thousands of scientists from varied disciplines in both the public and private sectors worldwide. One of the greatest impacts of having the HG sequence may well be enabling a radically new approach to biological research. In the past, researchers studied one or few genes at a time. With whole genome sequences and new high throughput technologies, we can approach questions systematically and on a much broader scale. They can study all the genes in a genome, for example, all the transcripts in a particular tissue or organ or tumor, or how tens of thousands of genes and proteins work together in interconnected networks, or orchestrate the chemistry of life. So basically, it's trying to show क्या हमारे पास फायदे हैं, क्यों हमने जीन सीक्वेंसिंग किया. सो मेनली it will help us to understand the human genome in a better sense, the human genetic material, to identify the disorders, to see how we can improve ourselves, or any of the sequencing. We can study the biological systems in a well coordinated manner, because now the scientists have everything. They have the technology, they have the sequence, and now they can work on it. Coming to a very important topic, that is DNA fingerprinting. As stated in the preceding section, 99.9% of base sequence among humans is the same. almost same है. Assuming that human genome as 3 into 10 raised power 9 base pairs, and how many base sequences would there be differences? It is these differences in sequence of DNA which make every individual unique in their phenotypic appearance. हर किसी की unique phenotypic appearance है क्योंकि हम सब में genetic material differ करता है. If one aims to find out genetic differences between two individuals or among individuals of a population, sequencing the DNA every time would be a daunting and expensive task. Imagine trying to compare two sets of 3 into 10 raised power 6 base pairs, हाँ, क्योंकि हम diploids हैं. DNA fingerprint is a very quick way to compare the DNA sequences of any two individuals. हम दो individuals का DNA sequence अगर हम उसको compare करें तब fingerprinting बहुत easily हम use कर सकते हैं, because you cannot afford to every time check and demarcate between the DNA sequences of two organisms. It's a tedious job to do, right? All right. DNA fingerprinting involves identifying differences in some specific regions in DNA called as repetitive DNA. तो हमारे में specific sequences होते हैं, repetitive DNA होता है which is specific to every organism, and that is where we look into the difference. Fine, because in these sequences a small stretch of DNA is repeated many times. ठीक है, बहुत सारे repetitive units मिलेंगे. These repetitive DNA are separated from bulk genomic DNA as different peaks during density gradient centrifugation. ये हमें different peaks पे मिलता है, हम centrifuge करते हैं इनको. The bulk DNA forms a major peak, obviously, and the other small peaks are referred to as satellite DNA. Fine, एक हमारे पास bulk DNA होगा और हमारे पास satellite DNA. Depending on base composition, AT rich or GC rich, length of segment and number of repetitive units, the DNA is classified into many categories such as microsatellites, mini satellites, etc. तो वो हम देखते हैं based upon various factors. These sequences normally do not code for any protein, but they form a large portion of human genome. Protein तो नहीं बनाएंगे लेकिन actually human genome का बहुत बड़ा हिस्सा होते हैं. These sequences show high degree of polymorphism and form the basis of DNA fingerprinting. Since DNA from every tissue such as blood, hair, follicle, skin, bone, saliva, sperm, etc. from an individual show the same degree of polymorphism, they become very useful identification tool in forensic applications. Further, as the polymorphisms are inheritable from parents to children, DNA fingerprinting is the basis of paternity testing in case of disputes. तो सिर्फ इतना ही नहीं कि हम लोग forensic sciences में use कर सकते हैं इसको कि भाई दो organisms है तो culprit कौन है, हम उसका repetitive DNA sequence check करेंगे because it's organism specific, but also for paternity testing, say who is the father of the child if there are certain disputes. Fine, so that also we can do from this only, from DNA fingerprinting, because these this polymorphism, this is inherited from the parents to the children. Fine, और ये हर टिश्यू में प्रेजेंट है. As polymorphism in DNA sequence is the basis of genetic mapping of human genome as well as DNA fingerprinting, it is essential that we understand what DNA polymorphism means in simple terms. तो हम तब से करें polymorphism वो fingerprinting में help करेगा, genetic mapping में help करेगा. What is it? Polymorphism means variation at genetic level arises due to mutations. Polymorphism, various appearances. Fine, so genetic level पे क्या variation आ रहा है, and we know how does genetic variation take place. It's only because of mutations, right? How we evolve and stuff. We've already done that. New mutations may arise in an individual either in somatic cells or in the germ cells that generate gametes in sexually reproducing organisms. ठीक है, कहीं पे भी mutation हो सकता है. If a germ cell mutation does not seriously impair individual's ability to have offspring who can transmit the mutation, it can spread to the other members of population through sexual reproduction. Allelic sequence variation has traditionally been described as a DNA polymorphism if more than one variant or allele at a locus occurs in human population with a frequency greater than 0.01 in simple. अच्छा, अब क्या हो रहा है? Allelic frequency change होता है ना, we know there's a dominant allele, there's a recessive allele, there's polymorphism at a certain locus. Fine, even that is polymorphism. In simple terms, in an inheritable mutation, if an inheritable mutation is observed in a population at high frequency, it is referred to as DNA polymorphism. ठीक है, कोई ऐसा म्यूटेशन है जो बहुत हाई फ्रीक्वेंसी पे देख रहे हैं उसको हम DNA polymorphism बोलेंगे. Simple. The probability of such variation is to be observed in non-coding DNA sequence would be higher as mutations in these sequences may not have any immediate effect or impact in an individual's reproductive ability. ठीक है, जो non-coding DNA sequence होगा वो उनमें ज़्यादा म्यूटेशंस हो सकते हैं, उनका कोई इफेक्ट नहीं होता है हमारी एबिलिटीज़ पे. These mutations keep on accumulating generation after generation and from one of the bases of variability or polymorphism. There's a variety of different types of polymorphisms ranging from single nucleotide chain to very large scale changes. For evolution and speciation, such polymorphisms play a very important role and you'll study these in detail at higher classes. तो जब ये वेरिएशंस कलेक्ट हो गए, accumulate हो गए, eventually हम evolve हो गए और polymorphism came into being. Why? Because there was variations generation after generation. The technique of DNA fingerprinting was initially developed by Jeffries. So who's the father of DNA fingerprinting? It's Alec Jeffries. Fine. He used a satellite DNA as a probe that shows very high degree of polymorphism. What is a probe now? Basically, DNA probes, they're stretches of single-stranded DNA and they're used to detect the presence of complementary nucleic acids. ठीक है, जो हमारी target sequences होती है ना, तो by hybridization you'll tend to find the कॉम्प्लीमेंटैरिटी की बेसिस पे हम दूसरे वाले का जो न्यूक्लिक एसिड है उसकी कॉम्प्लीमेंटैरिटी चेक कर सकते हैं. इट्स बेसिकली अ सिंगल स्ट्रैंडेड डीएनए स्ट्रेच. फाइन, दैट इज अ डीएनए प्रोब that shows very high degree of polymorphism. It was called as variable number of tandem repeats, VNTRs. The technique is used earlier involved southern blot hybridization using radio labeled VNTR as a probe. अब इसमें क्या क्या था? Isolation of the DNA, सबसे पहले then digestion of DNA by restriction endonuclease, separation of DNA fragments by electrophoresis, gel electrophoresis, we've done this before, transferring, blotting of separated DNA fragments to synthetic membranes such as nitrocellulose or nylon, hybridization using labeled VNTR probe, and detection of hybridized DNA fragments by autoradiography. A schematic representation of DNA fingerprinting is shown. The VNTR belongs to a class of satellite DNA referred to as mini satellites. So mini satellites भी it's a certain amount of DNA, ठीक है, जिनको हमने centrifugation में classify किया था, तो VNTR भी mini satellites को ही belong करता है. A small DNA sequence is arranged tandemly in many copy numbers. The copy number varies from chromosome to chromosome in an individual. The number of repeat show very high degree of polymorphism. As a result, the size of VNTR varies in size from 0.1 to 20 kb. Consequently, after hybridization with VNTR probe, अब हमने एक probe लिया हुआ है, जब उसके साथ हम hybridize करा रहे हैं, the autoradiogram gives many bands of different sizes. These bands give a characteristic pattern of an individual DNA. It differs from individual to individual in a population except in the case of monozygotic or identical twins. सिर्फ जो monozygotic identical twins होते हैं, उनमें सिर्फ DNA fingerprinting, in an individual, it'll be the same. The sensitivity of the technique has been increased by use of polymerase chain reaction, PCR. Consequently, DNA from a single cell is enough to perform DNA fingerprinting analysis in addition to application in forensic science. It has much wider applications such as in determining population and genetic diversities. Currently, many different probes are used to generate DNA fingerprints. So if we try and recapitulate all that we did in DNA fingerprinting, basically, ये किसने ये method devise किया था? It was Alec Jeffries. After he noticed that certain sequences of highly variable DNA, जिनको हम mini satellites बोलते हैं, they do not contribute to the function of gene, but they're repeated within the genes. Fine. Then he recognized that each individual will have a unique pattern of these mini satellites. Fine. जिसमें the only exception was the monozygotic twins, जैसे identical twins हमारे पास होते हैं. Fine. तो सबसे पहले we'll collect a sample of cells, blood, skin, hair, whatever. They'll contain the DNA. DNA is then extracted from the cells and it is purified. फिर उसके बाद what will we do is we'll be using enzymes, fine, which are going to break this DNA into different stretches, into different fragments. Then the enzymes produce fragments of varying lengths that were stored by placing them on a gel, ठीक है, and then we'll subject them to gel electrophoresis current के through. फिर जो shorter fragment होगा, the more quickly it'll move toward the opposite pole, anode, we know it already. The sorted double-stranded DNA fragments, they were then subjected to a blotting technique, ठीक है, जिसमें हम उनको क्या कर रहे थे? We were splitting them into single strands and they were transferred to a nylon sheet. तो blotting में क्या कर रहे थे? हम splitting कर रहे थे double-stranded DNA की और nylon sheet पे उनको transfer कर रहे थे. The fragments underwent autoradiography in which they were exposed to DNA probes. अब क्या करेंगे? ऑटोरेडियोग्राफी में डीएनए प्रोब्स ले ली हमने, विच वाज सिंथेटिक, जो हमने जो बाउंड, दे वर बाउंड टू द मिनी सैटेलाइट्स. A piece of then the X-ray film that was then exposed to the fragments and a dark mark was produced at any point, where a radioactive probe had become attached. वहाँ पे dark सा हमें एक mark सा दिखता था और जो resultant pattern आता था that could be analyzed and you could see कि eventually probes जो है वो binding में काम आते हैं. They're the stretches of single-stranded DNA जो complementary basis पे क्या करते हैं? They are going to bind to our DNA जो हम उसको यहाँ से दे रहे हैं. Fine. So that is a probe. Probes भी हम synthetically ही बनाते थे. Eventually last में radiography के उसके बाद हम एक्सरे की वजह से एक्सरे फिल्म ले लेते थे, ठीक है, उससे हमें एक डार्क मार्क मिलता था वेरेवर अ रेडियो एक्टिव प्रोब हैड बिकम अटैच्ड टू द गिवन पीस ऑफ डीएनए, जो हमने एक्सट्रैक्ट किया था हमारे जहाँ पे भी फोरेंसिक साइंसेज या जहाँ पे भी हमारा टारगेट साइट था वहाँ से हमने लिया हुआ था. सो दिस वाज जस्ट अ just like a recapitulation. And also you have to remember that this approach was also then later on हमने इसको बहुत we could modify it in a sense that we used polymerase chain reaction, ठीक है, PCR technique लिया. Basically it will amplify the desired fragment of DNA जो हमें चाहिए, ठीक है, many times over, so you'll create thousands of copies of the fragment. That is what we did, and I think that's it. That will complete our lesson. So thank you so much for watching. I just hope it was helpful for you, and keep watching. Thank you.